Thank you for joining us today and giving us your time. I know it's after lunch. If you have coffee, that's great. Probably it's good time to have coffee as well. Or we will finish off your day slowly as well with a little bit of sharing of the knowledge and interaction. I hope for some interaction today as well. So as I mentioned before, feel free to unmute yourself if you have any questions. But today the topic will be from edits to insights and how custom CRISPR cell models are rewriting research. As Sharon mentioned, we collaborate together with Labospace to support you in terms of logistics. But if you have any technical questions, I am always here to support you and introduce you to what we can offer and how we can support your research. So I want to start with positioning us in terms of, doing the research and what is actually important when it comes to making sure that your research is successful. And usually that comes from how we actually plan our experiments and how we plan to expand on them potentially. And there is a simple example over here showing, I think, the biggest challenge that we currently face when it comes to doing research successfully. The first thing is that we have a lot of data coming from AI tools at the moment. AI drug discovery is a very trendy topic, but also, GWAS studies that were done, they allowed us to see hundreds, thousands of different edits or different point mutations that could be related to different diseases. But then let's say that we study one disease. We found 100 potential targets that we could look at, but what we do normally is we use our rational filter. And this rational filter is completely normal. I had it when I was a scientist myself. I stepped away about 5 years ago, or more, actually, no, 8 years ago from the bench. But I had that myself as well, that I was doing around 5 models maybe. So that was my iteration, essentially, that was going through any of the experiments. And then we know that, like, when it comes to drug discovery, every single step kind of dilute the success. So discovery going all the way to clinic. 5 models with that attrition rate of drug discovery will essentially statistically give me maybe one gene to study that would be important for my disease. And then as an outcome, essentially, like, if I were to push it all over to the clinical trials later on and maybe collaborate with pharma, then we would launch 0.01 drug on the market, which is not enough. Obviously, this is not a good number. And that same comes with any kind of research success rate, right? So if we start with small number of models, we identify less genes, we have a smaller impact, less things to publish as well. So what if we could actually work a little bit different and utilize all of those US studies? What if we could use all of the AI delivered cell models and do the comprehensive screening, do hundreds of cell models, then identify potentially 50 genes or more that could be relevant to the disease, and then essentially pushing it, of course, all the way to the clinical trials, we could launch drug actually on the market in a successful rate. And I wouldn't be here as well if it wasn't possible. Of course, like, it's always due to like your time frame, your budgets, and how you want to do your research, and every research is slightly different. But I just want to show you that this just imagine is not just imagine anymore. We can actually support you with doing a more comprehensive studies. So that's why I'm here. And here's a EditCo in a nutshell, if I were to say. If I were to put everything that we do on one slide, that's essentially what it is. And you can see we are divided into pots. One is do-it-yourself CRISPR. So that's where you can get the gene knockout kits, you can get array screening libraries. So essentially tools to do CRISPR yourself. So that's one part of our portfolio. But the second part of the portfolio is custom CRISPR cell models that can be done for you. So in this case, you don't have to worry about designing your experiment as CRISPR. You don't have to design the guides. You don't have to look for the donor template if it's point mutations and things like that. You literally tell us that you want to work with immortalized cells, iPSCs, or human primary cells, and what type of edit you want to do. But we will go through it later on as well. I'll keep it here, and we can share this slide later with you. Everything is here clickable, so you can go into any of the specific part of that workflow and look more into it and see more details. But when you look at the slide, I grayed out these two parts because that's when, like, is it the knock-ins? And I think that maybe we don't give enough justice with that slide here for the knock-ins and what can be done. So I'll have a separate part of the conversation specific to knock-ins and what kind of tools you can use to expedite your research. But when you look at majority of those products over here, they're actually knockouts. And it's still something that we see being a trend, that knockouts is the first kind of leg into cell modeling and doing custom cell models by our customers or with us when they outsource. So I think it's important to mention how we perform it and why the tools that we use are superior to other tools that you may see on the market. Uh, and main thing when it comes to our knockouts is really utilizing this XDL multi-guide technology. You can see it in all of our products in the do-it-yourself tools, but also in the cell models that we do for you. So I want to touch on what actually the XDL technology is and why is it working so well. So essentially what XDL technology is either you can order the cells from us with already knocked out cells, with already knocked out genes of your choice using that technology, or you get essentially the multi-guide strategy that this technology utilizes to do your research or to do your cell model yourself. The way it works is that it has up to 3 good guides working in one early exon. So here is like a distribution how it would roughly look. It would never be the first one because we might have some alternate start site in that first exon, so we want to avoid that. But essentially, will be one of the first exons that we will target, and then we'll have 3 cut sites, essentially 3 different cut sites of those guide RNAs. So we have 1, 2, 3 here. But not only they create the insertion deletion, right? So insertion deletion is a common way of how essentially we generate the knockouts. We introduce the frame shift, and that's how we knock out the genes. We actually aim for something more than this, and we aim for the very controlled fragment deletion. So they're spatially coordinated in order to introduce that fragment deletion as part of knockout. So this fragment deletion can happen between the cut site of guide 1 and 2, 2 and 3, and 3 and 1. So there are 3 options for that fragment deletion to happen. And what that allows us to do is that actually in our hands, even with the cell lines like T cells, we can go up to 100% knockout efficiency at the heterogeneous pool stage. So just after transfection, we don't do the clonal isolation, we still can go up to 100% editing efficiency, which allows you to work earlier with your cell models. And you have really, really good results from that one as well. And as I mentioned, it targets one early exon. So if you were to do it yourself using that technology, you just run one PCR reaction for genotyping, and then you upload it into, like, you do the Sanger sequencing or NGS. If you do Sanger, you can upload it into our free software called ICE, and then you can get the data from this one PCR reaction. So you don't have to worry about amplifying even more. And all of essentially the guides are pre-designed for majority of the genes in the whole human genome and whole mouse genome, and they are ready to order. So it's very fast turnaround time, and then you have also the guarantee of the success. So as I mentioned before, we have 2 options for this multi-guide design if you wanted to do it yourself. One is a gene knockout kit, and essentially you just select the gene of your choice, and then you order the kit, which is ready for you to arrive within a week of time. We currently have a 50% discount for the new users, so feel free to reach out to Sharon if you were interested in trying it. So your first kit of 1.5 nanomole multi-guide strategy will be essentially 50% off using that coupon code. But then we started the conversation talking about doing more than one gene at the time or one alteration at the time. That's when we can think about doing something a bit bigger, which also works out better in terms of costing, actually, and that's doing array CRISPR screening library. So if you have 20 genes or more, then I would recommend you going with the array screening library versus the single gene because it works out better cost-wise. But from 20 genes all the way to the whole human genome, you can order the array CRISPR screening library. You can choose one of the pathways that we have. So if you study, for example, like G proteins, or if you study, I don't know, like like druggable, for example, genes, currently druggable genes. We have those kind of libraries. I think we have like 50 different ones that you can choose from, but you can also create your own. So you can give us the list or even populate it on our website. If you go to order now, you can literally go and select everything that you want, and you can order the library based on your needs. Based on the volumes, based on the layout as well. So I always say if you wanted to have a heart shape here instead of, like just doing one by one, you can do that. It's completely up to you. Valentine's is coming, so maybe heart shape design could work here. But what makes it so powerful is that we all of the multi-guide designs are actually already pre-designed and sitting in that compound. Think about it as a big fridge which has compartments for each of those vials inside of it. Then whenever you order a library from us, it will be synthesized for you. So even if you choose one of our pathway, it's not something that was sitting on the shelf for God knows how long. We actually will be generating that library fresh for you, and it takes about a week for us to do. And it's so fast because essentially like you can see a little pipe over here. This pipe shoots the vials through the ceiling to land on this machine over here, which is essentially a robot that populates your plate of your choice. So 96 or 384 well plate, depending on the volumes you're working with. And that's essentially the closed system that allows it to be fully pre-designed, like designed for you and ready to ship whenever we're done with the fresh material. And I just want to show you that even if you expand from working with one cell to the library that I mentioned to you, it's not something that we say that's going to work and you don't have any data to prove that. Well, we have the data from the UCSF. This is the data that was generated at our customer site with their cells. They were working with primary dendritic cells. And you can see that they got 97 million editing efficiency, and that was assessed via NGS. And essentially, you have 280 different genes in that library. The way to look at it is if it has a color, not gray, that means that it was successful editing. There will be some genes which may be essential, and that's why they are grayed out. But on average, if it's like a normal gene, you should get a very good editing efficiency. And across all of those genes, you can see that this editing efficiency was achieved. So if you work one vial at a time, so one gene at a time, or if you work with the library, the XDEL technology should really give you the best results that there are on the market. And they achieved 97% median. I saw recently the data coming from the big pharma that they had 100% across all of the genes. So it's always down to the optimization. Are there any questions to that part in the chat, or anybody would like to ask anything? I'll have a moment to sip. Sharon, let me know if there's anything.
There is no
questions for now?
No questions.
Okay, no worries. Sometimes it's good. Maybe it was very well explained. I don't know. All right. So like then the big question comes in. What if I actually don't have capacity or expertise to do CRISPR myself? And I think some of you may be in that situation at the moment, or sometimes you have the expertise and you have the time, but you just want to focus on doing something else rather than developing CRISPR yourself. And I think it's very fair because even when you think about cell culture media now, back in the days we were doing cell culture media ourselves. We were optimizing the pH, we were buying reagents separately, but it was taking us a very long time to get all of the reagents we needed, adjust them correctly, and then making like there was no guarantee that it will work every single time, right? So every time the batch will be slightly different. Now majority of us order the cell culture media directly from vendor with the guarantees that it's going to behave well, right? So if you think about it, that's what we think CRISPR is heading towards, that it shouldn't be an experiment of its own. It's just a tool for you to do the further research, right? So CRISPR cell line is a starting point for you to make discoveries. So you may not want to spend your time and expertise to do that yourself. And actually, it's not even, one thing is like doing it yourself, but also it actually is not so simple to do it, especially when you do it for the first time. And there are 3 numbers here coming from the benchmark report that we've done back in the day. And I just want to show you what's behind them because I think that's the biggest struggles that people report, even experienced people working with CRISPR. So 43 is the average percentage editing efficiency that people get, right? So across the board, knock-ins, knockouts that they do, 43% at the pull stage is roughly what they experience. 7 is a number of attempts that they take in order to get to those 43%. So, it's not only you do it once, you do it at least 7 times to get roughly 43% on average if you do it yourself. And the biggest number here is 100. And I think the one that resonates with me personally the most is 100 hours is a number of hands-on time that it takes per clone to be developed in your hands. And we are not calculating, of course, the incubation times and the time that you are, like just away from the lab. It's your actual manual work. And this is quite common. So if you experience something like this, don't panic. Like this is completely normal. It's just a process that we need to go through if you want to do CRISPR yourself. But as I mentioned, you don't need to do it yourself because you can get access to EditCo's CRISPR cell line engineering lab, which looks like this. So you can see it over here. We can do both knockouts and knock-ins on that platform. We work in parallel. So whenever you have one or hundreds of edits you want to do, we will start and finish them at the same time, right? So there is no lagging in terms of delivering the cell models for you. And where this like automation come into play the most is when it comes to optimization of the cell lines and being efficient. And something that you will see doing CRISPR yourself, when you see that one cell line is working better than the other, when you thought you have the best protocol for transfection, and then you don't get the right result, it's completely normal because there are different types of cells react which are easier or harder to transfect. So back in the day, since the girl which we came out of, we're running 200 point optimizations, meaning different reagent concentration and ratios in order to optimize each of the cell line that we were working with. Now we have over 1,400 cell lines that we have already optimized, and if you wanted to get the list of cell lines we can work with, I can share it with you later on. But I want to show you like what we found is that there will be some cells that are easy to optimize that you do it like 7 times and you get 90% editing efficiency almost. There will be the moderate samples, but there will be also samples that are very, very challenging to edit. And even us, for hundreds of point optimization steps, we couldn't get it to work normally. And 7% is actually something that people always say that they get roughly 7 to 10% as an editing efficiency at the pull stage, which is not ideal because isolating clones from something like this is very hard. So you may need to run, like, I don't know, hundreds of them in order to find one that works. We turn it a bit around. And with those optimizations, we managed to achieve 82% editing efficiency of the knockout in the THP1 cells. So I don't know if any of you ever work with them, but THP1s are suspension cells which are difficult to edit. We got to 82% compared to the standard 7%. That's why sometimes outsourcing can be a game changer when it comes to doing things. Because sometimes, even with the THP1s, if you do something like a target validation, 80% is more than enough for you to see the effect of the knockout, right? So you don't have to go all the way to the clones, which takes 4 months usually to get to if you grow from the single cell. You can get that in like 4 weeks time, right? In 4 weeks time, you're ready to go and you're ready for the publication. So you can be way ahead of the game, especially when it comes to the knockouts. So just to clarify, like when it comes to our platform, that the platform covers the second one that I showed you, all of the custom CRISPR cell models that we were doing. And of course, not majority, but many of them are the knockouts that we do in immortalized iPSCs or human primary cells. But we actually see the more interest recently even in the knock-ins. So that's something that I wanted to now focus on, just as I'm not going to talk about knockouts apart from what we've talked about so far, I just want to make sure that you're aware that we can do the knockouts still on the platform, both pools and clones if needed. But I want to share with you a bit more data on the knock-ins because I think that's a very interesting topic. Because when it comes to knock-ins, there's almost limitless options of what can be done. We can start with something simple like point mutations or SNVs for disease modeling. Then we can introduce something like luminescence or fluorescent tag into your gene of interest directly endogenously in your gene at the C or N terminus. Same we can do with the degradation tag. So something that can, rather than work with like doing a knockout, you can degrade the protein. So you work at the protein level, not at gene level, which can be very crucial for essential genes, for example. Then you can do something like affinity tags that can be used for the detection or purification, or you can go even higher to do the reporter or fusion genes between 1 to 3 kilobase pairs, which can be used for biosensors. So there are so many different applications. If you have any specific application that will be interesting for you, feel free to reach out. We can talk about it and see what's feasible for you. If you don't have an idea at the moment, also feel free to reach out because there might be something that we can find already from what we've done in the past that could work specifically for your work as well. But I want to give you a bit of intro of what can be done and how we see what we see the most popular now from our customers' requests. So I want to start with the luminescence and fluorescent tags. So this will be again tagged endogenously, and there are many applications that you can do. You can work with GFP or other common tags, but I want to highlight to you the tag that I'm not sure if you've heard about it before, but it's called HYBID. Hybrid tag is a technology which is powered by Promiga. So it's a technology coming from a company called Promiga, but we can deliver it to you in any cell type that you want. So essentially we will tag the protein, the protein of your choice, but like essentially we will work at the gene level. So we will work at the C or N terminus of your gene. And then we will tag it and then will be translated into the protein. It's very small protein tag. It's just 11 amino acids, so it doesn't change the biology, and you can choose which part to tag essentially. Then essentially what happens is that it reunites with the large bit to create a very bright and luminescent glow. And it's very, very, very bright compared to GFP or anything else. So it's very easy to be detected and it's very sensitive. So even small changes in the protein levels can be detected. So where can it be used? So protein expression and quantification, as I mentioned, it's ultra sensitive and it works in real time. So you can measure the protein levels in cells or in lysates. So it doesn't have to be only in the cells. Protein localization and trafficking, that's what it can be used as well because you can track the surface expression, internalization of the protein, or secretion of the proteins into cytoplasm and stuff. Then protein stability and degradation studies. So you can monitor the turnover or the halftime of the protein. And another thing is when you do any drug screening, if you have any compounds that may be affecting protein expression or localization of the protein, that's exactly what it can be used. So you have the data before adding the compound and after adding the compound, and you see exactly what's happening with the proteins that you study. And as I mentioned again, it's tagged endogenously, so it's very, very precise. Then to give it even more power, you can combine it with some other tags. And the most powerful combination that we saw is combining hybrid with a degradation tag. So essentially the way it works is that we have a gene of interest, we tag either C or N terminus of your gene with the hybrid and the degradation tag, which is essentially the tag, that's the tag name. And then what you can do is you can keep it as is and nothing will be happening. Or you can start adding the substrate into the cell culture media, and then the substrate will be joining the degradation tag, and it will be triggering the degradation of the protein that you study. So think about it like you have your protein here, you have your tag, just like tagged essentially. It's always going to be in the protein. When you start adding the compound, it attracts the ubiquitination system, and then it gets into the proteasome and it disappears. The gene still will keep on working, but the protein level will be changing and it acts very fast. So the first thing to look at it is like the amount of compound we would be adding. And the compounds, by the way, are commercially available, so you don't have to worry much about it, and you can read more about it. And if you are interested, I can share more information with you. But essentially, the compound that you like, the level of the compound that you're adding will trigger the different levels of degradations. So that's one thing. And the beautiful thing is that this is the data from the luminescence. So you can see how sensitive it is, right? Because you can clearly see the levels of the degradation with the luminescence assay. If you don't have access to the luminescence assay, you still can use HiBit because there are antibodies against HiBit. So then you can do something like a Western blot, and you can see clearly like the same kind of interactions or the same level of degradation as you see in the luminescence assay. So that's, first is control of the compound or amount of compound you need to add to achieve your downregulation or like your degradation of the protein to the levels you need for your assay. But then also you can see how fast it acts. So within 3 hours, it's usually ready for your assay and it will maintain within the cell culture for a while, but it is reversible. So if you stop adding the compound into your cell culture medium, it will spike back again because the gene will still keep on producing the protein of your choice. So again, it can be used, you can use the Western blot to track it and see how fast, for example, is your gene, like how fast does it take for the gene to produce your protein of choice, for example, because you will see the peak going up as well. So if you have access to luminescence, that's great because you can see it real time. If you don't, Western blot will work just as well. And where this combination of HiBit and degradation tag is very powerful. Couple of them here. Knockout alternative. So let's say that you study an essential gene that you know that if you knock it out, it will cause a cell death, right? You cannot really do anything. We cannot knock it out. But there might be a way to work with this system so you can control the degradation level and see which one is lethal, right? Or you can sometimes go away with just doing your experiment while this protein is not present within the gene because you can just introduce the degradation just before the experiment. You can also study the acute protein degradation with simultaneous monitoring. You can target the validation. You can use it for target validation and drug discovery, high throughput screening. It can be used also to studying the protein turnover and stability, as mentioned, mechanistic studies for downstream signaling, or even in vivo monitoring of protein degradation, because there is data, there are papers showing that it was used also in mice. So you can then see clearly, like when you add the compound and that, for example, tumor is disappearing in the mice, things like that. So I think it's a very beautiful system that I'm not sure how many of you were aware of. If you wanted to hear more about it, let me know. But I want to finish off with saying why would it be worth a while partnering with EditCo for these kind of assays, both knockouts and knock-ins. First is that we are very precise and reproducible. So our edits have average editing efficiency for the knockout of 98%. That's at pool stage. If you wanted a clone, it of course will be 100%, and you will get 2 clones and 2 vials each as a deliverable, but it will take longer. It will be slightly more expensive. For the knock-ins, smaller knock-ins, we have 50% editing efficiency, and our viability across the passages is now over 90%. So we are very gentle on the cells with the high editing efficiency maintaining. We have a very rapid turnaround times. It's for the knockouts is 3 to 4 weeks only. For clones is 8 to 10 weeks. And the reason why it's so long is because we would really take the pool that takes 3 to 4 weeks. Then we would do the single cell printing, and then we would grow the clones from single cell. That's the time that it takes essentially here. That's the time to growing the cells into the confluency and then making sure that you have 1 million cells per vial, more or less, depending on the cell type. And then for the knock-ins, it's slightly longer, but still, that's as fast as we can go because essentially we will start the project as soon as we get the order. There is no delay on our platform. We will start the day we get the project and we will ship it to you as soon as the cell grow. So that's usually the time that it takes for the cells to grow from the single cell. It's highly scalable. So as I mentioned, everything is working on the automated platform, and every edit is running in parallel. So, if you wanted to do hundreds, thousands of cell models, knock-ins and knockouts at the same time, we can definitely do that for you. And we have access to over 1,000 cell lines that you can choose from. Some of them are free of charge that we can just source for your research, so you don't have to worry about sourcing them either. That will be for the iPSCs, immortalized cells, and for the T cells as well. And it's very cost efficient. So actually, if you were interested in comparing how much it costs to do CRISPR yourself versus how much it costs for us to do it for you, with all of the expertise that we have and the success rate that we have, it is actually very, very cost effective, because many times when we do the calculation of how much it costs us to do CRISPR, we don't take under account, all of the reagents and the bulk reagents that we need to get, how much time it takes us to get all of them, all of the repetition we need to go through to get to anything. And then, of course, our salaries and everything is also included, I think, and time that we spend, especially in the lab, is included in that cost calculation. So I will be more than happy to share that if you are interested too. And these are my contact details. If you want to connect with me on LinkedIn, here is a QR code. You can scan it now and join my network. I'm sharing also our website here, and there is my contact detail. If you wanted to talk about any very technical aspect of today's talk, or if you have any ideas, just feel free to drop me an email. We can book a meeting. We can meet together with Sharon, so we can streamline the whole workflow for you. But if you have technical questions, I'm here essentially to help you. And I'm just going to leave it open for the questions. So coming back to the overview slide, if there was anything here that was interesting for you, but I didn't touch on, feel free to ask. Or if there was anything during the talk that was interesting, or if you have any questions, just let me know. So I'll leave it open to you guys. Let me know if there is anything.
Thank you, Zofia. Any question?
Yes, please. Hello.
Yeah, hi.
Hi. Thank you. Very nice presentation. I want to ask if you have any experience, or if you can offer the service of doing CRISPR in vivo.
Yeah, so that's tricky. We offer, so the only in vivo version of what we do would be when we do the cell line, for example. Is it mouse that you're interested in?
Yeah.
Okay. So if you wanted to work with like mouse samples, we could edit the mouse cells, and then you could inject the mice with those cells. That's as far as we can go with in vivo. Because when it comes to editing mouse itself, it will require the lentiviral editing or stuff like that. We work with RNP complexes, so we work on the cell level.
Okay.
But we had customers working with mice with our cell lines that we developed for them.
Okay, I understand.
Mm-hmm.
No, actually, we already knocked in the cell lines, but we are interested in an oval origin of cells, and as we know, up to our knowledge, there's no oval melanocytes, murine healthy cells available. So we thought about doing the CRISPR maybe in vivo directly.
Yeah, so that probably you would need to work with viruses in order to do that, and that's not something that we can offer at this point. We focus on the RNP complexes because we just saw the data, and when it comes to drug discovery, usually that's where we are sitting in. So that's why I think.
Okay. Another question, maybe you mentioned it, but I lost. For knockout, you use how many gRNA you use guide RNA for a gene?
Yeah, so we use, like, I can go back here. Let me just actually go out and I'll show you. So we use up to 3.
Up to 3.
So,
okay.
That's how our kits work. So we have up to 3. I'm saying up to 3. That will be accurate for 95% of the genes. In some cases, like around 5% maybe or less, we'll have less than 3 guides, so it will be 2 usually. Because we have very strict criteria of how we select those guides. We want them to be spatially coordinated, and we don't accept any off targets or mismatches, right? So that's why some of them will have 2 guides, but majority of them have 3 guides.